So hello everyone, welcome back to again my channel. So today in this video we talk about the structure of RNA. What are the composition of the RNA? How many types of RNA? Okay, all we discuss. Generally in this video we talk about the mRNA. Okay, so let's start. So let's start from the beginning. Okay, so on the previous video we will discuss about the structure of DNA, right? What are the properties of DNA? We all know, all we understand, but here we talk about the something different. Okay, we all know the DNA, so we all know the DNA is a double-stranded structure, right? That means it's composed of two strands, right? But so today we discuss about the single strand. So that is called the RNA. Okay, so right here the RNA. It is similar like DNA. It is one type of nucleic acid. It also nucleic acid means it's also composed of so many type of nucleotide. Okay, different type of nucleotide joined one by one to form a chain, and that's why it is called the nucleotide. Okay, so here the RNA full form is, we all know, I write here nucleic acid, ribonucleic acid. Okay, RNA full form, same like DNA. That's mean here it is also consists of nucleotide. Nucleotide means we all know that it actually consists of some type of nitrogen base. Okay, and second one is, we all know, the phosphoric acid, that means phosphate. Another one is, that is sugar. Sugar means here we use the ribose sugar. Okay, here that it is formed, actually it is made up of ribose sugar. That's why it's named the ribonucleic acid. Same like DNA. Okay, so actually basically the RNA are single strand. Okay, so how it's look like? It's look like here too. Okay, generally what happened? The RNA is single strand, but to form the double strand, it's formed a hair loop structure. Okay, why it's formed the hair loop? Because of the formation of a bond. Okay, actually, what is the first we have to understand what is the basic difference between the RNA and DNA. Okay, I say right here the RNA and DNA. Okay, all we understand. We all know that DNA is a double strand. I just write here double strand. Okay, but here the RNA is only the single strand. Okay, all are same. Okay, second one is, in case of DNA, we all know they have the nitrogen base, that is the purine and pyrimidine, right? But here, in case of pyrimidine, some change in RNA. In case of DNA, we all know the adenine, guanine, purine, right? And thymine, cytosine present in pyrimidine, right? But here, in this case, that means in case of RNA, they have, I just write here shortly, adenine and guanine. In case of purine, but here one is different. In case of pyrimidine, in the DNA they have the thymine, right? But here in case RNA, instead of thymine, they have uracil. Okay, and both are same. We all know in the DNA thymine also present, but here uracil also be present. Okay, so how it looks like, if suppose this sequence A G C G C A, if such type of sequence also present, in the opposite we all know, in case of DNA, adenine binds with the what? Thymine, right? Adenine binds with thymine and guanine binds with the cytosine. But here, in case of RNA, adenine binds with the uracil, okay, and guanine binds the cytosine, same like DNA. So if the adenine also present in the one strand, in the opposite sequence they have also present the uracil. If the G also present here, here also present the cytosine. If it is cytosine, here also present G. In the same line, the sequence arranged in a rapid manner. That means here simple things is that if the one sequence adenine also present, in opposite they are also present the uracil instead of thymine. Clear? Okay. That is the actual difference between the DNA and RNA. Already clear? Okay. Now we have to understand, sir, how many types of RNA. Okay? Okay, so I erase all those things. Okay? Okay. Now we have to understand how many types of RNA. Okay. Generally, RNA, I write here the RNA. RNA are basically three types. Okay. First one is called mRNA. Second one is rRNA. And third one is tRNA. They all have the different meaning. Okay. Now we have to understand what is the actual meaning. So first we goes to the mRNA. mRNA generally is a huge type of RNA, and the M means here M means messenger. Okay, do you use messenger? We always use Facebook Messenger, WhatsApp Messenger. Message means what? It's also transform the data, or you like to say information, from one region to another region, right? In the different in the world, right? So in the same way, the RNA is such type of particles which carry the information from one cell to another cell. It carry the genetic information from one cell to another cell. So that's why it's called, and this type of RNA is called the messenger RNA. Okay, just simply understand. Okay, next one is the RNA. R means ribosomal RNA. Okay, ribosomal. Ribose such type of small RNA. Actually, naturally, rRNA also used for the formation of a ribosome. For the production of the ribosome, we generally use the rRNA. Okay, and this is the one type of RNA, tRNA. tRNA is called the transfer RNA. Transfer, which act as a carrier to transfer anything. Generally, if I give an example, suppose we use different loaded lorry, right? Loaded truck to carry the goods from one region to another region throughout the country, right? So in the same way, within the cell, from suppose this is one type of cell, this is one type of cell, to carry any information, act as a carrier for the protein synthesis. You all know that RNA also used for the protein synthesis, right? So they also use the tRNA. Okay. So all we understand. So generally in this video we talk about the mRNA in details. Okay. So I erase all those things. Now we talk about the mRNA. Okay. So here you will also see the structure of mRNA. This is the structure of mRNA. I write here the mRNA. Okay, mRNA. mRNA basically general RNA. We all know the central dogma rules, right? We all know central dogma rules. We all know. I also tell you before, from the DNA to DNA is called, we all know, the replication, right? From the DNA we also produce the mRNA, that is called transcription. This technique is called transcription, we all know, right? From the DNA also formed, right? We all know. Again, from the mRNA, DNA also formed. This is called reverse transcription. Right? We all generally know, right? And from the RNA, we can produce the protein. That is called translation. And that's all events generally happen in our cell naturally, right? And it is called the rules of central dogma. We all know, right? I just write here for the revision. Okay, so that is the whole process always run throughout our cell, right? But here, so if we need some protein, so first we have to produce what? mRNA. Why? Because from the mRNA we can get protein, right? So how the mRNA form? So mRNA form generally form through the base transcription. That is, in that case we need DNA, right? So we all know where the DNA actually is situated. DNA actually situated within the nucleus of our cell, right? So if I draw a cell, right? Within the cell, generally this is the nucleus. Suppose this is the nucleus. So here, so many another cell organelles also present, which also float within the cell, right? So generally the replication process happen within the nucleus, right? So we all know during the cell cycle, from the one cell, when the new cell also formed, first the nucleus also divide, then the cell outer structure also divide, right? So during the nucleus divide, the nucleus become changed, right? So in that time, the DNA, right? DNA also will replicate, right? DNA can be copied, right? So in that case, in this case, when the DNA also be copied, in that time, it also carry the information to the next cell. So how is transferred to the next cell? The information. How the information from the one cell transferred to another cell? How? It's also held through the RNA. So basically the mRNA acts as a messenger which also carry the information from the one cell to another cell, or you like to say from the cell nucleus it goes to the cytoplasm region, right? And then the cytoplasm is transferred to the near beside another cell, right? Then who acts as a carrier? Here the carrier is also called the mRNA or messenger RNA. Now we have to understand, sir, what are the structure of mRNA. Okay? So let's start from the beginning. So I use all those things for the better understanding. Okay. So first mRNA. Okay. So mRNA are generally basically the large RNA. Okay. So it have some region, specific region. Just you see. So basically, suppose this is a DNA. How it's formed, I just say shortly. Suppose this is a DNA. Okay. From the DNA, we have to produce the RNA. And we all know the RNA is the single strand, right? So here, so how it's formed, right? Single strand from the double strand is from the single strand. That means we have to remove this double strand, right? In case of DNA, right? So here, the RNA formed. When the RNA actually formed, and from the RNA we will have to produce protein, right? If generally we all know, generally in the world two types of cells are generally there, right? One is eukaryote another is prokaryote, right? In case of eukaryote means in case of us or in case of plant, they are more advanced. Here in this case, they produce from the one mRNA, they produce one protein. From the one RNA, they also produce one protein. Okay, in case of eukaryote. So that's why they are also called monocystonic mRNA. Okay, mRNA. So which one is monocystonic? If from the one RNA, one protein also from, it is generally seen in eukaryote type of plants or animals, and that is called, they are called, this type of RNA also called the monocystonic RNA, or what we say monocystonic mRNA. And but in case of prokaryote like bacteria, okay, they have the ability from the mRNA or from the RNA, they can produce so many proteins. These are the proteins. They can produce so many proteins from the one RNA. So that's why they also called polycystonic. Clear? In case of prokaryote, from the one mRNA, they can produce multiple type of protein. So that's why they are called polycystonic in case of prokaryote. But in case of us or in case of eukaryotes, from the one RNA, they can produce one protein during their translation. Okay. So that's why they are also called the monocystonic RNA. Clear? Okay. Now we have to understand what are the actual sequence of a mRNA. So actually, when the mRNA also formed from the during the transcription, what happened in the mRNA? They form a cap-like region. It's the same. It is same like DNA. We all know that DNA have the 1-5' region and the involve 3' region, and due to its constant double stranded, they have also the complementary sequence at the end, right? In the same line, because of the RNA is single strand, that's why it have only one 5' and another one is one 3'. Okay? So just you have to understand, you have to remember that in case of RNA, they have the ability to join with the other sequence. Because in case of DNA, we all know that DNA have the double strand. So here, actually at the end of the 5' region, it has the tendency to join with the another sequence. So that's why this region have a cap, okay, to protect the sequence, to protect the end part. In the same way, in case of RNA, okay, it have the cap region. Okay, it have the cap region, and this cap region have some group that is called methyl group, and this methyl group obstruct to join with the other sequence. That means it also protect the RNA to join with the other sequence. Okay, it is same like us. We all know to from the any protection we use the helmet, right? During the riding, right? In same way, RNA also protect themselves to use a cap, and this cap region is called the methylation region, or methyl group also present in cap region, and that's why here it does not recognize any sequence. That means it is also block the other sequence to join here. Okay, so that's why it is called the cap region. Clear? The cap region? Cap region actually present at the head of the mRNA. Clear? Okay. Then I also discuss about the non-coding region. First we have to understand what is the coding. I had the coding. Coding means what? Suppose this is a one type of strand. Okay. So there are so many bases are there. Just I write here in a frequently. Suppose this is a one type of sequence. Okay. So here three bases. Suppose this is a three base. So three base consists to form a consists to form a character, and that's why it is called a code, or you like to coding sequence. This is a sequence. This is one type of sequence, right? So here three nucleotides joined by each other to form a sequence, and that sequence is called the coding sequence. Okay, and coding sequence means it carries some character, and somehow if any sequence does not carry any character, and that region is called the non-coding sequence. So within the mRNA, after the capping region, they have also the non-coding sequence. It is actually 250 to 100 and consists of 100 nucleotides. Okay, 50 to 100 nucleotides also carry. Clear? Okay, this is a non-coding. That means it does not have any recognition site to join any sequence. Okay, this is a just non-coding sequence because it does not carry any character. Okay, and is also called the intron. Okay, just let's say intron. Clear? Okay. After the non-coding sequence, there have some sequence that is called initiation sequence. Now you have your question in your mind, sir, why it is called initiation? Initiation means, or you like to say, in this area it have some sequence or some coding sequence. Suppose this is a subtype of code. This is called AUG. These three type of bases, right? Three type of bases to form a code. That means it's also carry the some character. Okay, we all know from the mRNA we may produce protein, right? So here to formation of protein we can use the mRNA. So when the translation start, in that time we use the ribosome for the protein synthesis. Okay, during translation. So in there when the translation start, in that time when the ribosome bind with the mRNA, the translation start from the starting region. There is a specific region, and that region is called the AUG coding region. If the AUG codon is present in the mRNA, from that region translation start. Okay, from that region translation start. To recognize this site, the translation actually start. Okay, so that's why it's called initiation sequence. Initiation sequence means the region, or we like to say the point from where the translation made to produce protein. Okay, so that's why it's called initiation sequence. And this sequence also present within the mRNA after the non-coding sequence. Okay, so this is the one type of coding sequence, and this is specific for the RNA. All case of protein, all type of protein may start formation from that sequence. That is the AUG. Okay. Okay. After the coding sequence, this is the initial sequence. Okay. Then the normal coding sequence also start. This is the normal coding sequence means here so many another code also present, but all have the character, right? What I say, coding means it also carry the character, right? So here this coding sequence have the nucleotide that's all carry the character or information. So that's why it's also called the exon. Okay, right here exon. Exon means the functional part. That means the region of the mRNA from where the actual functional protein actually formed. So that's why it's also called the exon. Clear? Okay. So when the actual protein protein formation also be finished, after the finishing of the protein formation, there is another codon also present, that is the non-coding region, or also say the stop codon region. Okay, stop codon. So there the stop codon are the UAA, UAG or UGA. This is the stop coding. Stop coding means during the translation we have to need to finish the protein formation, right? So there is some sequence in the region present which also block the protein synthesis, right? So here in that case, to remove the ribosome, actually what happened, suppose this is a mRNA, within the mRNA ribosome also be present. When the protein also be make things start, in that case the ribosome join at the AUG region, that is in that region, AUG. And when the ribosome also be moved side by side, protein also be formed. After the protein formation will stop when the some another non-coding sequence also be present, and that is called UAA UAG. Is also be specific. Okay, and in that case the ribosome also be removed and protein also be formation also be finished. So that's why it is called the non-coding sequence, also called this codon is called the stop coding. Stop coding region. Okay, it may be UAA or UAG or UGA. It is actually specific for in every mRNA. Okay. So when the stop coding also be formed, stop coding, after the stop coding, there is another region that is called the poly-A tail. Okay, this is the poly-A tail. Poly-A tail means here the adenine base also present in a repeated manner. Now the question arises in your mind, sir, why the poly-A tail also be there? Actually, it is present to to protect the stop coding region. Actually this formation, we all know the cytodogma rules from DNA to RNA, RNA to protein, and all events happen in a few microseconds, right? So in that case, generally what happened, somehow the misfield also be there. So to avoid from the misfield, they can protect RNA to form a poly-A tail. Okay, and it's also help to recognize from the RNA to DNA formation. On the previous video we will discuss the RT-PCR, right? In the RT-PCR we will discuss how we form DNA from the RNA using the poly-A tail sequence, right? In that video I also discussed there, right? So that's why in somehow from the RNA we have to make DNA. In that case the poly-A, the specific sequence also be held. Okay, so this is the actual phenomena of the structure of RNA, mRNA. I think all you actually understand. If you have any doubt, you can comment on my box. So that's all for today. We will be back on the next video.